This video will teach you about an interesting part of genetics called epistasis. Before I begin to explain what epistasis is, let's go over some of the aspects of genetics you may need to proceed. An allele is an altered form of a gene. In a typical human childbearing family, the variation of the gene comes from both the mother and the father's genes. A genotype is the mix of alleles that a person normally cannot see, as the genetic material is microscopic. This can both be homozygous and heterozygous. Homozygous is represented by two of the same letter, either both capital or both lowercase. Heterozygous is represented by two varying letters, one capital and one lowercase. A phenotype is how the gene is expressed. You can normally see this expression with the naked eye, as it displays a physical characteristic. For example, different eye colors are genetically connected to the parents of the person in question, and eye color is easily seen on humans. Epistasis is the interaction of two different genes, where one gene controls the phenotype of another gene. Note that it is only the phenotype that is affected and not the genotype. Lastly, in our genetic lingo, we have dominant and recessive. Traditionally, a dominant gene only needs one capital letter to mask over the other phenotype, but it can have two capital letters and have the same result. A recessive needs two lowercase homozygous letters to mask over the phenotype. With dominant and recessive epistasis, there's an inhibitor or a factor that prevents another step to take place towards a phenotypic product. Let's use cereal as our example. The creators of this batch of cereal started with 2 Cheerios in the first generation. When bred, genetically, the Cheerios could have 16 different outcomes for their baby cereal. Both of the original Cheerios are heterozygous for color, which means they both have a capital and lowercase representative letter. The 2 Cheerios have a heterozygous capital A, capital B, lowercase b genotype. Remember that inhibitor I mentioned? Let's name it inhibitor G, which is going to be a piece of candy for our purposes. The candy is the dominant A in the genotype. If candy was in the Cheerio's genetic makeup, then it stays looking like a regular Cheerio. Without candy being added, the Cheerio can end up an Applejack because the candy is what prevents the Cheerio from absorbing the color, which changes it into the Applejack. The food coloring added can represent the letter B of the genotype. If one of the B's are dominant, the color will change to look like an Applejack. Epistasis will always create a ratio. As you can see here, we have 13 Cheerios to 3 Applejacks. The Applejacks are created because of the dominant B.